Alright, hello everybody. I hope you're all having a wonderful evening. So yesterday we discussed the phases of mitosis, which now we realize produces more somatic cells in organisms. So today we are going to be looking at another process called meiosis. And the process of meiosis produces our reproductive cells. And reproductive cells are what give rise to new organisms. So hopefully you're beginning to see the differences between meiosis and mitosis, and hopefully you'll have a clearer idea of these differences after we have gone through each of the phases of meiosis. So let's take a little bit closer look at these phases. So like mitosis, there is prophase, metaphase, anaphase, and telophase. But one major difference is that meiosis involves two rounds of these phases, called meiosis 1 in yellow and meiosis 2 in orange. Each phase in meiosis 1 and 2 are labeled quite nicely as either prophase, metaphase, anaphase, telophase, with a corresponding 1 or 2 at the end, just to make it a little bit easier for us. And realize that this diagram is not as labeled as much as it was in the mitosis diagram, but hopefully you are beginning to recognize the different things that we have seen before in the cell. Alright, so let's begin. So at interphase. We know that mitosis involves somatic cells, but what are the cells involved in meiosis? Do you think it would be more somatic cells? Well, no, because then how would our cells know when to undergo mitosis and when to undergo meiosis? So meiosis involves different types of cells, and these cells are called germ cells. Germ cells, like somatic cells, proceed through the growth and synthesis phase of interphase before dividing. Remember that chromosomes are replicated during this S phase of interphase. So therefore, at the start of meiosis, a germ cell contains duplicated chromosomes, and each chromosome is made up of a pair of identical sister chromatids held together at the centromere. So understand that by convention, each pair of sister chromatids is considered to be one single chromosome as long as the chromatids remain joined at the centromere. So hopefully you will understand these terms. And you'll understand that difference. Alright, so now we're going to begin looking at meiosis 1. So meiosis 1 starts with prophase 1. And in prophase 1, each pair of homologous chromosomes align themselves side by side. And this process is what we call synapsis. These four chromatids synapse together is what we call a tetrad. And a tetrad is a pair of homologous chromosomes that are synapsed together, and therefore we have one pair of sister chromatid paired with the homologous sister paired chromosome. So this next part is going to be a little bit tricky to understand, but just bear with me. I'll explain it a little bit better in the next diagram. So any chromatid on one pair of sister chromatids is non-sister chromatids with any chromatid on the other pair of sister chromatids. So we'll look at that a little bit more clearly in this diagram here. So along with this synapse process, things that occur in prophase of mitosis are very similar to what occurs in prophase 1, including the spindle fibers lengthening, the centrioles moving to opposite sides of the cell, and the nuclear membrane disappearing, which causes the release of the chromosomes into the cytoplasm. Alright, so, I just kind of noticed that our centrioles have changed color. They went from purple to yellow. It's just a little bit strange. Alright, so here we have metaphase 1. And first I'm going to discuss that idea of that non-sister chromatid. So if this is a pair of sister chromatid, and this is another pair of sister chromatids, and these two chromosomes here are homologous chromosomes. So one sister chromatid on this pair is non-sister chromatids with a chromatid on this chromosome here. So we could say that this chromatid is non-sister chromatids with this chromosome or this chromatid. Okay? So sister chromatids non-sister chromatids, or non-sister chromatids, or non-sister chromatids. Okay? Realize it doesn't have to be the one that's right next to it. It can be non-sister chromatids with this one as well. Alright, so in metaphase 1, the spindle fibers guide each tetrad to the center of the cell. However, note that the chromosomes do not line up side by side as they did in mitosis. Instead, they line up in their homologous pairs. So for each tetrad, one homologous chromosome faces one pole, and the other homologous chromosome faces the opposite pole. And see how this spindle fiber attaches to this centromere, and this spindle fiber attaches to this centromere. Alright, so now we'll move on to anaphase 1. So the spindle fibers begin to shorten, and this causes the homologous chromosomes to separate from each other and move to opposite sides of the cell. Since the sister chromatids are still intact, the centromeres do not split apart. Alright, so in telophase 1, just note that some cells actually skip this phase, and they also skip the phase prophase 2. But some cells actually go through these two phases, so we'll make sure we go through what happens in these phases. So in telophase 1, the chromosomes begin to uncoil, the spindle fibers disappear, and a nuclear membrane forms around each set of chromosomes. The cytoplasm also divides, which is what we now know is called cytokinesis, and two cells are formed. Each of these new cells contain one set of sister chromatids, and is now considered to be haploid, since the homologous chromosomes are no longer together in one cell. However, we still have two chromatids forming that sister chromatid pair, right? We still have that pair of sister chromatids here. So in order for these cells to be reproductive cells, because they're not reproductive cells yet, each new cell needs to undergo meiosis 2. And if it didn't, otherwise new organisms that were made would have doubled the DNA, because we still have that pair of sister chromatids. So we have to go through meiosis 2 in order to become reproductive cells. And also be aware that chromosome replication does not take place before entering the next phase of meiosis. So no chromosome replication happens before entering meiosis 2. Alright, so now we're going to look at meiosis 2. And meiosis 2 is pretty much the exact same process as mitosis. However, I'm still going to go through each phase just so you can have a better review, and it kind of reviews mitosis as well. Alright, so in prophase 2, the chromatin condenses into chromosomes, the nuclear membrane disappears, and the centrioles move to opposite sides of the poles, or opposite sides of the cell. In metaphase 2, the spindle fibers attach to the centromeres from each pole. They attach to the centromeres and guide them to the equator of the cells. In anaphase 2, the spindle fibers begin to shorten, which causes the centromeres to split apart from each other, and also causes the pair of sister chromatids to split apart from each other, moving to opposite poles of the cell. In telophase 2, the chromosomes begin to uncoil, the nuclear membranes form around each set of chromosomes, and you can see cytokinesis occurs, which splits the cytoplasm. And by the end of meiosis 2, now we have 4 daughter haploid reproductive cells. which are now ready to fuse with other reproductive cells to create new organisms on Earth. Alright, so I can already kind of see some confusion arising for some students on this fact. So each cell that enters meiosis II is haploid, but these cells have replicated chromosomes, which is our sister chromatids, our pair of sister chromatids. So by the end of meiosis II, the daughter cells are still haploid, but now they contain a single unreplicated chromosome. So if you think about it, this unreplicated chromosome is homologous to an unreplicated chromosome in another haploid reproductive cell. This is why every human has 46 chromosomes, or in other words, 23 pairs. So there are two questions I want you to address after watching this presentation. And you will answer these questions on the website through a poll tool. So the first one, I want you to tell me, what is the diploid number of this particular organism that we see here in this diagram? So remember that a diploid number is represented as 2n. So in humans, our diploid number is 2n equals 46. So the second question, let's take a look back at metaphase 1. Did you notice anything that was a little strange in this phase that I didn't discuss? I didn't discuss this color switching here, alright? And the reason why is because I want to talk more about this in tomorrow's class. So what do you think is going on here? Did the cell make a mistake? Did the people who created this entire diagram make a mistake? Or if you don't think it's a mistake, please answer in the poll other. And if you do answer other, be prepared to share your ideas in tomorrow's class. Alright, so that is it for today. Remember to check out everything on the website for today's lesson, and don't forget to answer the two poll questions. And have a wonderful evening, and I will see you all tomorrow. Bye-bye!